Hello everyone, it's me, Jerez and my partner, Campos. Welcome to our presentation titled of Adenovirus Vectors, the anatomy of genetic delivery system. Today we are going to explore how science has transformed the most common virus into a powerful medical tool.
To understand gene therapy, we first need to understand the fundamental delivery problem. We have a message: the therapeutic DNA or RNA designed to code the fully cellular process. However, this naked generic material degrades rapidly in the bloodstream and cannot penetrate cell membranes on its own. Therefore, we need an envelope or vector, a microscopic delivery truck engineered to navigate the body, breaks the target cells, and safely deposit the payload in the nucleus.
What better delivery truck than a virus? Nature has perfected viruses over millions of years to be highly efficient at injecting genetic instruction into human cells. The process has three steps. First, we start with the wild-type virus. The second, then comes the disarmament, where scientists remove the viral genes responsible for causing disease and uncontrolled replication. Finally, we get the vector harmless purpose to carry a therapeutic genetic payload safely.
Let's look at the profile of our vector today, the adenovirus. It belongs to the adenovirus family. Its architecture is fascinating because it is a naked virus with no lipid envelope, protected by an icosahedral capsid measuring 70 to 90 nanometers in diameter. In humans, there are 57 serotypes, but serotypes 2 and 5 are the most frequently used for vectors. In nature, its primary targets are the epithelial cells of the respiratory tract, eye, and gastrointestinal tract.
If we look closely at its anatomy, we see a design perfect for delivery. First, he has a targeting system made up of the fiber and node domain, which are producing spikes that mediate initial attachment to host cells receptor like CAR or CD46. Next is the penetrant base, which acts as branching chairs, to interact with the host cells integrins to trigger endocytosis and internalization. The headstone serves as the cargo hold, housing the genetic material. And finally, the payload, the linear double-stranded DNA flanked by inverted terminal repeats and packaging signal.
A critical characteristic of this vector is that its DNA does not integrate into the host genome. Unlike retroviruses, the adenoviral genome remains inside the nucleus as an independent, circularized fragment or episome. The great benefit of this is that it eliminates the risk of intersectional mutagenesis, meaning we don't run the risk of inadvertently activating an oncogene. The trade-off is that because it doesn't integrate, the therapeutic gene is not copied when the cell divides, leading to transient expressions.
Okay, adenovirus has involved in the lab. Third generation vector with the A1 region lead could carry about 7 at 8 kilobase. Second generation vector increased this to about 14 kilobase. But the major breakthrough is the third generation, or goalless vectors, devoid of all coding viral regions. This high-capacity adenovirus can accommodate massive payloads of up to 36 kilobytes of DNA, allowing the delivery of exceptionally large genes.
Using this tool comes with pros and cons. Its enormous advantages include its massive payload capacity, high efficiency in infecting both dividing and quiescent cells, rapid high-level expressions, and the fact that it is easy to purify and concentrate to massive scales. However, its cons are significant. They trigger high immunogenicity, strong innate and adaptive immune responses. Humans often have persistent immunity that neutralizes the vector, and the expression is transient, typically lasting only 5 to 10 days.
This brings us to what we call the immunity paradox. We know the immune system is the primary limitation, but it is exactly what defines its most powerful medical application. If the goal is lifelong replacement of missing proteins in a chronic genetic disease, the immune system destruction of the vector after 10 days is a failure. But if the goal is to train the immune system or destroy a specific target, that intense, transient inflammatory response is the ultimate weapon.
The best example of this is vaccines. In the global response to COVID-19, we saw adenoviral vectors shines with vaccines like AstraZeneca and J&J. Setups involve exploiting the vector with DNA encoding a target antigen, like the spike glycoprotein. The vector then infects our cells, turning temporarily into highly efficient antigen factories. The strong natural immunogenicity of the adenovirus acts as a powerful adjuvant, triggering robust T cells and B cells responses. Finally, the body rapidly clears the vector, leaving behind long-term immune memory.
Another amazing application is the cancer treatment. Adenovirus can be engineered as conditionally replicate adenovirus rats here with E1A mutation to replicate only inside cancer cells. When the virus enters a healthy cell, missing regulatory genes prevents it from replicating, and the healthy cells survive on your. But when it enters a tumor cells with broken metabolic pathway, the virus hijacks the cells, replicates widely, induce cells death, like tumor lysis, and new viral particles are released to infect newborn tumors.
As we have seen, the adenovirus acts as a genomic escaper. It offers unmatched delivery from transference layers, complex generic payloads in the nucleus of cells. It saves episomal action experts high-level generic control without permanently altering the host chromosomal blueprint. And finally, while high immunogenicity prevents lifelong single dose course, it perfectly positions the adenovirus as a primary tool for vaccines and target cancer eradication.
To conclude, covering our presentation directly, in the landscape of genetic medicine, the adenovirus is not a subtle instrument. It is a powerful, highly delivery system, mastering the balance between viral engineering and human immunity. So that's it. Thank you so much for your attention. Bye.